thank you everybody for joining us for No Time Like the Presentation Part 2. So if you weren't with us this morning, here is what's going on. We had scientists, postdocs, and graduate students submit 90-second videos explaining their work. And myself and 7 other scientists and folks who have nothing to do with science all watched those videos and picked the 10 that were the easiest to understand and the most engaging. And now we're hearing 10-minute presentations from 10 of those scientists, and this is part 2, and these are the scientists that are going to be telling us more about their stuff, and we're so excited. This whole program is run by Skype a Scientist. We are a nonprofit organization that tries to bring science to as many people as humanly possible, and this is all part of that. So here's how this is going to work. All of us are going to turn off our cameras and turn off our microphones with the exception of the person talking. So this is the order that you're going to go in. Catriona is going first, and then Nicole, then Catherine, then Taryn, then Vitica. And I think that's it for me. The only other thing, if you appreciate what we're doing here at Skype a Scientist, you can support us at patreon.com/SkypeAScientist. And really that's it. So I think it's time to get started. So I'm going to hide because I am not the star of the show, the scientists are, and gonna have our first person take it away.
Hopefully you can see me and my screen. Okie dokie.
When we go outside, we like to put on sunscreen. But what if once you rubbed it off, there was a rash underneath?
Hi, so my name is Catriona, and a couple of years ago, I had an allergic reaction to sunscreen. So today I'm going to share with you my story, and it's a bit of a personal story as well as one of scientific discovery. And it all begins with the immune system, which evolved to fight. It fights viruses, bacteria, and parasites, but it can also fight things that are essentially harmless, but it perceives as a threat anyway. And when the immune system overreacts like this, that's resulting in allergy. So central to the immune system are T cells, and these are the snipers of the immune system. I call them snipers because they take their time to set up, but once they do, they're really good at their specific targets, like snipers. We have killer T cells that kill infected cells to limit the spread of infection. And a little bit topical, you can think of it a bit like quarantine. So if you kill anything that's already infected, you keep the healthy cells safe. And we also have helper T cells, and these coordinate the immune response and send alarm signals to other immune cells. Now, all of our T cells are responding to molecules, and most of them react to broken down proteins called peptides, but they're not important because I'm more interested in T cells that react to fats and oils. So what they're seeing are small molecules inside fats and oils called lipids. Now, these T cells are a little bit different from conventional T cells, so I like to think of them as hipster T cells. So they're not conventional, but I think they're pretty important. T cells need molecules to be presented to them on a silver platter, otherwise they won't be able to see the molecules. And to this end, we have antigen-presenting cells, or I'll refer to them today as Santas. The Santas will present molecules for T cells to see. And they'll pick up lipid molecules and wrap them up in a gift wrap called CD1A. And once a Santa has picked up lipids, wrapped it up in CD1A, it then presents its presence to T cells. And any T cell that reacts to this is called a CD1A reactive T cell, or CAT cell. And like I said, they're hipsters. We don't know too much about them because not many people study them, but what I've found so far is they're a little bit more like the helpers. So all of our T cells, when we're growing up, need to learn the difference between what's self and what's non-self, because we want them to attack invading bacteria and viruses, but we don't want them to start attacking our own bodies. So they all have specially shaped hands that help them with this. So that when we're presenting fat from our own bodies or lipids from our own bodies, the shape of the cat hand doesn't quite fit it. So we don't get activation of the cat cells. But if we're picking up lipids from an invading bacteria and bacteria are covered in unique fats, this time the shape fits. So the cat cell becomes activated and attacks the bacteria. This can also happen with poison ivy, and depending on where you are in the world, you'll be very familiar with it or not. So it's a plant that when you touch, you can get red rashes. And again, if we're presenting the oils in poison ivy, the shape has the right kind of fit and the cat cell becomes activated. But sometimes this line between what's self and what's non-self gets a little bit blurred. So cat cells can play a role in allergies to bee and wasp venom. What happens is that once you're stung, the bee and wasp venom breaks down your own skin oils. So we all have a lot of oils in our skin, some people maybe more than others, and that's fine. But if the oils start to look a little bit different to normal, they're presented to the immune system and it can't be recognized as self anymore. So cat cells attack, they recruit all their immune cell buddies, and we get an allergy as they start to attack the skin thinking it's infected when it's really not. So why would we react to our own bodies? It all comes down to shape. So if you think about the CD1A and lipid shaking hands with a cat cell. Now I know you probably can't shake hands at the moment because we're all on lockdown, but if you were to shake someone's hand and they were holding a small coin, you'd still be able to kind of touch hands and shake. But if one of you was to hold a large bulky phone, you can't really grab the person's hand anymore and shake it. And the same thing goes for cat cells. So with the cat cell hand, it can come in nicely and snug around small lipids and touch both the lipids and the CD1A, so it becomes activated. But if the lipid is large and bulky, it sort of gets in the way and hinders that interaction. So it's only when lipids have the right shape as presented, that's when we get cat cells reacting. So to summarize this, lipids are picked up by CD1A and they're presented by SANTers or antigen presenting cells to cat cells. And if the cat cell has the right kind of shape to fit around that lipid, then it activates the cat cells. But sometimes cat cells can turn against us. So as I mentioned, I did have an allergic reaction to sunscreen a couple of years ago. This is me. And a normal person would have never used that sunscreen again, and that would be the end. But I'm an immunology student and a bit of a nerd. So I thought, oh, it's a tea. What is it? I did some detective work to work out what was the ingredient in that skincare product or that sunscreen that I hadn't used before. And I found that one of them had the right kind of shape to fit into CD1A, and it's called AB oil. So my first question was, does AB oil activate cat cells? It was all very coincidental. So to test this out, I grow cat cells together with SANTers, or antigen presenting cells, and CD1A in a dish, and I add AB oil. And yes, indeed, the cat cells become activated. So somehow this AB oil is being taken up and activating cat cells. But what I want to know is it AB oil presented as it is, or is it being broken down by the time it goes through our skin and gets to cat cells? So to test this, I'm looking only now at CD1A and lipids, so we can forget Santa for a moment. CD1A is presented in a production cell, a little bit like a factory, and everything it wraps up in CD1A are self-lipids because it has to work with what it's got. If we're thinking about the elves making and wrapping presents for Santa, they can only wrap up the toys that they have in the factory. And we can look at the identity, the unique identity of all these lipids, and there's, under normal conditions, quite a variety. You've got large, bulky ones, you've got small ones. But I wondered what happens if I add AB oil. Is it broken down? Is it kept as is? So now what I found was that a lot of the CD1A had AB oil inside. So now looking at this, it seems like AB oil is kicking out all of the normal lipids in favor of itself. So perhaps AB oil can indeed cause allergy, which is completely coincidental for me. What we think is happening is that in the skin, we have antigen-presenting cells, or SANTs, and they usually just present self-lipids because that's all they've got. They work with the oils in the skin. But when we're adding this skincare product, it has AB oil in it, and that kicks out all of the self-lipids. So now we're presenting AB oil instead, and the cat cell comes along and becomes activated. So now we've actually found that there are a couple of oils in skincare products that can cause allergy, and these are found in deodorants, moisturizers, sunscreens, etc. So it really is important to find out what's going on so that we can stop it, because now I'm allergic to some skincare products, but yay, science. We really shouldn't have to think twice about putting on sunscreen. It's called skincare, not skin careful. And just because I've got a little bit of time left, I'm going to summarize this in a song. Patrolling your body are all these cat cells. They check the cells of your body to see if they're well. Looking for lipid molecules presented by CD1A. Attacking things not from yourself to keep bad bugs at bay. Cat cells attack. When they see something off, they call on immune cells, and the immune response kicks off. Cat cells are hipster T cells that recognize fat. When they see lipids from bacteria, they gear up for combat. Some recognize oils in sunscreen causing allergy. They mistake the oil for a threat and call the cavalry. Cat cells attack. When they see something off, they call on immune cells, and the immune response kicks off. So thanks for listening, and thanks to everyone who helped, and that's me.
That was amazing. Thank you so much. Okay, now it is time for some questions. Okay, in your assays, which for the general crew here means like tests, to see if your cat cells in your dish are activated, how do you know that they're activated?
That's a good question. So there are markers that immune cells express, especially T cells. So T cells express markers when they do become activated. So I'm essentially just testing to see whether the levels of the activation markers have gone up.
Cool, sounds good. And are cat cells only present in places that are fatty, or are they everywhere?
Mostly I'm looking for them in the blood and the skin, and they are present in both. It's a little bit difficult for us to say at the moment exactly everywhere in the body where they're found and where they're not found, because not many people are studying these cells. But no, they're not just only in fatty sort of places, which might actually be a good thing, because if they can sometimes accidentally be activated, you don't want them around a lot of fat.
Totally. Awesome. And then our last question, where did you get the cat cells that you were using in your study?
So sometimes they come from blood, and sometimes I engineer normal T cells. So they're sort of like the mainstream T cells that I was talking about. I engineer them to start becoming cat cells. So they're starting to express, or they're starting to show the kind of markers that recognize lipids instead of those sort of normal boring ones.
Awesome. Well, thank you so much. That was really, really cool. And also you have got to put that song on YouTube by itself, because that was, I've never heard a ukulele immunology song, and so that needs to be out there. All right, thank you so much. Next up, we've got, oh wait, before you go, is there where can we find you on the internet, and is there anything you want to plug?
Nothing I want to plug at the moment, but on Twitter I am at CatrianaNR. So you can see what I'm up to there. And I do write a couple of sciencey kind of songs. So if you follow me, you'll find them.
Well, we love that. Thank you so much. Okay, Nicole, you're up.
Okay. Hi, everyone. My name is Nicole, and I'm a graduate student at the University of California, Davis. I study the tiny cells that make up the human body. And inside these cells are special instructions called DNA. But when DNA is damaged, but when the DNA in some cells is damaged, a lot of diseases can arise. Researchers like me are using gene editing tools to try and understand these illnesses. And that's what I'm going to talk to you about today. I want to walk you through the basics of DNA and genetics in our cells and the tools of gene editing. And then after that, I'll bring all these concepts together to tell you about the specific problem that I'm researching. So with all that said, let's get started. DNA tells the cell how to make proteins, which are like little biological machines that do all sorts of work around the cell, like making energy from sugar and helping cells grow. We can imagine that our cells are like little biological Lego cities. There are tons of buildings and cars to make the city run, and those are the proteins. And each building or car needs instructions for how to put the bricks together and make them. And those are our genes. Put all of the instructions or genes together in one place, and that's the DNA of the cell. The cell reads the instructions or genes in the DNA to make proteins, just like you and I read the instructions in a Lego booklet to build a car. Now, I don't know about you, but I have definitely misread the Lego instructions before. I've put the brick in the wrong place or used the wrong one altogether. So instead of building this nice working car, I've made, well, this thing. And our cells can make the exact same mistake when reading our DNA, and that's what's called a mutation. Your cell misread the instructions, and now the proteins it produces either don't work correctly or work differently than they were supposed to. So when movies or TV shows make a reference to mutants or mutations, think like the X-Men or Teenage Mutant Ninja Turtles, that's exactly what they're talking about. So one of the tools that myself and other scientists can use to understand mutations is the gene editing tool CRISPR. CRISPR is a tool that researchers like me can program to cut open specific DNA sequences, make edits to them, and then change the genetic instructions in the cell. This in turn then changes how the proteins are made and overall cell function. So sticking with our Lego analogy, we have here DNA that is completely normal and unedited. Each Lego brick represents one step in the DNA instructions. We have 4 blue blocks and 4 yellow blocks. Now, along comes CRISPR, which in this case will be my hands, and it acts like a pair of scissors and cuts our DNA. Now, our cells have 2 options for how to fix this cut. One way is for it to try and put everything back together exactly the way that it was. The only problem is that because of some slightly complicated chemistry that we won't be able to get into here, the DNA can't just simply be glued back together. Instead, it actually has to take 1 or 2 blocks off first. Once it does this, it now has to try to remember exactly which blocks were used and how many. And I mean, to your cell's credit, a lot of the time, it gets this right. It's able to grab 1 blue, 1 yellow block, and glue everything back together. But sometimes it messes up and leaves off a block. So then you have 3 blue and 4 yellow. Or it adds an extra one, giving us 5 blue and 4 yellow. But either way, this majorly messes up the instructions. It would be like if someone ripped out the second page of your Lego booklet and still expected you to make the right thing. You can't, right? You can try and fill in the missing pages from memory or by guessing, but you're just not going to get it right every time. And the same goes for your cells. In this way, CRISPR can be used to disable certain proteins, and this allows us to study how the cell behaves when those proteins aren't around. Now, remember that I mentioned there is a second way to fix the cut CRISPR made. So the second way, which I'm going to call the copy and paste method, starts off like the first. We have our normal unedited DNA. CRISPR comes around, it cuts it, and we take off a couple of blocks. But this time, instead of the cell trying to remember what the original instructions were, we give it a little bit of help and give it brand new instructions to copy and paste into the DNA. So now, instead of 4 blue and 4 yellow blocks, we could have, say, 3 blue blocks, a red and a green, and 3 yellow blocks. And scientists can use this form of CRISPR to either fix an existing mutation or introduce a brand new one and learn how it affects the cell. So far, we have discussed how DNA holds all the instructions for cells to build, well, themselves, how errors in the DNA instructions are called mutations, and we saw that CRISPR is a gene editing tool that researchers like me use to make a snip in the DNA at a specific part of the instructions, and that the cell has one of 2 ways it can repair the cut that CRISPR makes. So now that we've laid all of that groundwork, I can finally put all these concepts together and tell you guys what my research is about and how I use CRISPR. So my work focuses on a type of protein called histones, which help the cell figure out which genes or parts of the DNA it needs to be following in the first place. But how do histone proteins help the cell figure out which part of the instructions they need to be reading? So one thing I haven't told you guys yet is that DNA is long, like really long. If you stretch the DNA from one single cell from end to end, it's about 6 feet. That's almost a whole foot taller than I am and just about as long as the average NBA player is tall. So since your body is made up of about 30 trillion microscopic cells and they each have 6 feet of DNA inside them, there needs to be a way to make all of this genetic information fit and humans not be as tall as skyscrapers. So your cells solve this problem by rolling up the DNA like we see in this diagram so that it's nice and compact, like when you roll up a piece of yarn into a ball. This process is called DNA compaction. And fun fact, when DNA is rolled up and compact like this, it's called a chromosome, which is this blue globby X here on the side. And histones help with that compaction. If we look at this diagram, the green strand is our DNA and it wraps around these blue spheres, which are the histone proteins. And when it does this, some instructions are easier for the cell and read correctly than others. Like this section here would probably be fairly easy for the cell to read. And this is perfectly normal. The cell has mechanisms to make the right part readable at the right time. Specifically, there are chemical groups, which are illustrated here as these blue arrows, that regulate how the histones end up grouping together. So when the histones are really bunched up like they are here at the top, those parts of the instructions aren't read and basically no protein is made. But when they are spread apart, like they are down here at the bottom, and the DNA, which is the green strand here, is actually exposed and the cell can see it, we make more proteins. Depending on how the DNA wraps around the histones and then how the histones group together, different parts of the DNA instructions will be read and followed by the cell. This is part of a process called gene regulation, which is where the cell controls which proteins and how much of them are made at any given time. So that's histones and compaction, both of which are really important for cell function. But just like any other protein, histones can also be mutated. And this is where my work comes in. When histones are mutated, there's a few different things that can go wrong. One of those is that these chemical groups, so the blue arrows, can no longer be added to the histones. So they can't control how bunched up or how loose the histones are. So this can result in histones being more bunched up than they should be, and so then we get less protein than we need, or other instructions being more open than they should be. So we make more of that protein than we should. And either way, the balance of proteins in the cell is messed up, and this can lead to all sorts of illnesses like cancer. So how does CRISPR relate to any of this? In order to better understand this mutation and the different effects it can have, I use CRISPR to make a break in the DNA and then have the cell use the copy and paste method we went over earlier to prepare that cut. The new instructions that give the cell replace the mutation or mistake that was in the gene that makes the histone protein. So now the cell will make the correct non-mutated version of the histones. And with this, I can study how the mutated histone is different from the correct one and how this changes how your cells read DNA. Okay, that's all I have. Thanks, guys.
Thank you so much. That's really cool. So you're not only like editing the DNA, you're editing the DNA of the thing that wraps up the DNA. That's like,
right, exactly.
That's awesome. Great, thank you so much. All right, so our first question, if you had the chance, would you alter your own DNA?
That's a really good question, and it's a hot topic in ethical debate. And personally, given my own experience with CRISPR, I wouldn't. It's not the easiest thing to make work exactly the way you want it to.
Very cool. So what diseases are associated with histones wrapping up the wrong thing at the wrong time?
Right, so histones, they help with development, so taking like a single cell human to being a full-grown adult, but I specifically study it in the context of pediatric brain cancers.
Awesome. Well, not awesome, very sad, but that's really important. Yeah. Thank you for answering. Okay, so if you know, human genetics is very complicated and we're still learning things all the time. And that's true of any animal's genetics. And so are there ever examples where you go in and you are thinking that you're making one change, but in fact, you make a different change, and what do you do in that situation?
Right, so that is something that most scientists have agreed, if you're using CRISPR, that you need to, if you make your edit, you then need to do basically this thing called whole genome sequencing. So go and read every single piece of DNA to make sure you didn't accidentally mess with a different gene. And it's a really important step of this whole gene editing process.
Awesome. Well, thank you so much. Where can we find you on the internet?
Right. So if anyone saw at the bottom corner of the PowerPoint, I have my Twitter handle. So it's Nicole, spelled NICHOLE_Lewis_. And then I just recently started up a YouTube channel called BioCorner, and there's 2 videos up right now.
Awesome. You've got great energy for a YouTube channel. That's really fantastic.
Oh, thank you.
All right. So thank you for sharing that with us. The next up, we've got Catherine.
Hey.
Hello.
Okay. Hi, I am Catherine Nugent, and I am a grad student at the University of Washington and a researcher at Lowell Observatory. I'm getting the screen share going. Okay, and I am going to talk about binary red supergiants in astronomy. And so first question, what is a red supergiant? Here is the constellation Orion, which hopefully many of you recognize. It has the 3 stars in the belt, which makes it pretty obvious, easiest one to recognize in the sky. And this star, Betelgeuse, in the armpit of Orion, is a red supergiant. And so this is the type of star that I'm going to be talking about today. And these are evolved massive stars. They are 10 to 30 times the mass of the sun, and they have been burning through their hydrogen and stopped burning hydrogen, progressed onto burning helium. They've now expanded and cooled down, and so they are thousands of times the radius of the sun. And so here we have a comparison between VY Canis Majoris, and the sun is just this tiny little dot next to it. And so if you were to put VY Canis Majoris in place of the sun in our solar system, red supergiants would extend out past Jupiter. And so they are huge stars. And to kind of put this in context, here we have what's called a Hertzsprung-Russell diagram. And on the x-axis, we have surface temperature with hot stars on the left and cool stars on the right. And then we have increasing luminosity, and so bright stars at the top and dimmer stars at the bottom. And here is our sun in the middle, just kind of a normal luminosity, normal surface temperature star. And these red supergiants are way over here in the upper right corner. They're incredibly luminous. They're some of the brightest stars in another galaxy, and they're incredibly cool, around 4,000 degrees Kelvin. And as I said, very, very large in radii. And so why do we care about red supergiants? They're pretty awesome stars, and at the end of their lifetimes, they explode as supernova. And so when they do this, they spend their lives burning through heavier and heavier elements, and then when they explode, they release all of this stuff into the interstellar medium. And that is actually a lot of the stuff that makes up us and our solar system. And so if you look at this periodic table, we see all of these little green elements are from exploding massive stars. And so we have some carbon, nitrogen, oxygen, aluminum. All of these things come from when the stars that I'm talking about, these red supergiants, explode and enrich the interstellar medium. And so for my work, I focus on binary red supergiants, and so stars that are red supergiants and have some sort of star that is kind of gravitationally bound with it. And when I started this research in around 2018, there weren't very many known, maybe around a dozen. And so my question was, are they not known because they're hard to find or because they don't exist? And so the main reason at this point was that they're really hard to find. And so generally when we look for a binary system, we look at what's called a light curve. And so we image the star over time and we look and see how the intensity changes. And so if it's a binary system, then it'll be bright when you see two stars, but then when the other companion goes in front, it'll block some of the light. And so the light will dip, and then it'll get brighter, and that process will repeat. This is also how some exoplanets are found. And so this works great if the star is kind of unvarying and is a constant source of light, but that is not the case with red supergiants. And so here we have a simulation. This is not an actual red supergiant. We can't see in this much detail because they're very far away, but this is a simulation of what the surface of a red supergiant looks like. And so we have these huge convective cells, and so all this gas is kind of coming up and it's cooling off as it gets a little further away from the star. That's the dark areas. And then as it comes back down closer, it's getting hotter and becoming light again. And each one of these is bigger than the size of our sun. And so these things are huge. And this video is it simulates around a year time span. And so if you were to try and take a light curve of this star, you would see that it's changing. It's not just kind of a constant light source. And so if you try and get a light curve of it, then it might even look like there's a binary companion because it's going to have these dips. And so this makes it difficult to find binaries around red supergiants using the common method of looking at light curves. And so when I approached this, first wanted to see what types of stars would be the companions to red supergiants. So say we have a red supergiant, would it have a star like our sun as a companion? Would it have another red supergiant? And so I used evolutionary models to determine that they would have these bright blue companions, B-type stars. And so they're very luminous, hot stars. And so I then went off searching for some red supergiants with bright blue companions. And to do this, I used what we call photometry. And so it's essentially taking images using different filters that show you light from different wavelengths. And so here is an image of a pretty galaxy, and in the one corner we have imaging from using ultraviolet light, and so we have filters that are only letting the ultraviolet light through, and then as you kind of go around you get to the infrared light. And so knowing the amount of light that is coming at you in these different wavelength regimes tells you something about the object. It could tell you what it's made out of, how hot it is, all different sorts of things. And so I used this method to try and find these red supergiant with the bright blue star companions. And so the general gist is that red supergiants, no surprise, are red. If you go outside and you look up and you're able to see Betelgeuse to your naked eye, it will appear quite red. And we can contrast this to Rigel, which is down in the bottom corner of Orion, and it is quite blue. And so these colors, they appear to us as you would see them with your naked eye. Red supergiants, red, bright blue stars are blue. So if you put them together, then the hypothesis was that you basically might find something kind of yellowish. And so it's a little more complex than that, but that's the general premise. And so here we have a plot that shows what the colors would be of these stars using different filters. And so what I want you to take away is that we have blue stars down here in the bottom left corner, and then we have these red supergiants that are single without a companion in kind of the upper right. And then all of this little black area is where the red supergiant with the bright blue star companions would fall in terms of color space. And so this means that, yes, they would look kind of yellower. And so we can then go and look for stars that have these colors. And so to do that, I went looking for stars in the Andromeda Galaxy M31, in Triangulum M33, as well as the Large and Small Magellanic Clouds. I went down to Chile in the southern hemisphere. And essentially what I did was I looked for, I used photometry to find a bunch of candidate red supergiant with bright blue star companion stars. And then I took spectra of the ones that I was really interested in. And I did that using this telescope right here. This is down at Las Campanas, one of the Magellan telescopes, 6.5 meter. And so spectroscopy, essentially you have a star, you put that light through a prism and it breaks it up into kind of, it stretches it out in terms of wavelength. And so you get what is seen on the right. We have on the x-axis, we have wavelength. And so we have bluer light on the left and redder light on the right. And then on the y-axis, we have flux. And so we can see how, what the star is emitting in terms of how much flux it's emitting as a function of wavelength. And these absorption lines that are labeled here tell us what type of material the star has. And so it actually tells us the kind of elements that make up the star. And so here is some spectra of some newly discovered red supergiant binaries that were located outside of our galaxy. So that's pretty cool, pretty far away. And so over here, where the red arrows are, shows that this is a red supergiant. These lines are what you would find in a red supergiant. They're titanium oxide. And then if we blow up this little region over here, then we see Balmer lines that are coming from the bright blue star. So spectroscopically, this is telling us that this is a red supergiant B-star binary. And so this was very successful. We found around 250 of these. And so I went from around a dozen known in the universe up to over 250 known in some of our surrounding galaxies. And so this was really cool. And so the next question is, so we have a red supergiant and a bright blue star. What happens if they get too close? And so here we have some images of modeling this. We have the red supergiant is played by my corgi Meg, and the B star by my cat Marlo. So they're gonna start off separated, but then gravity is going to pull them closer and closer together. So over time, they'll go closer. And then eventually, if the mass of the red supergiant is high enough, and so the gravity is high enough, they will actually merge. And so the red supergiant will consume the bright blue star. And when this happens, it'll speed up. And so it'll be rotating much faster than what you would expect. And so my research, my thesis is focused on trying to determine basically the number of binary red supergiants and then merged red supergiants based off of how fast they're spinning, and then single red supergiants. And this all kind of falls into answering the question of, how are all these heavier elements made? Because basically red supergiants are some of the stars that are making what we are. And there's the great Carl Sagan quote, "We are made of star stuff." And so thank you so much. I just want to have a quick shout out to my amazing collaborators, Emily Levesque, Phil Massey, and Nydia Morel. Feel free, here's my Twitter handle. Feel free to follow me on Twitter, and I am happy to answer any questions.
Thank you. This was very, very cool. Okay, we've got lots of questions here. One is, are there any community science projects that are at all related to astronomy that people could get involved in?
Yeah, the main one that I think of is always Galaxy Zoo. I forget if you Google Galaxy Zoo, it was one of the first kind of community-driven projects, at least that I heard of, and essentially it allows you to classify galaxies. And so you're very quickly trained on distinguishing between different types of galaxies, and just by clicking images, you're able to classify them, which it works better than just feeding it into a computer program in many cases. So yeah, it definitely allows citizen science work that leads directly to publications.
Awesome, thanks. So we know that Earth is currently in what's called the Goldilocks zone, so places where life can exist without getting totally fried or totally frozen. And so when a star is bigger, does that Goldilocks zone also get bigger? Does it get smaller? Like, is there any effect on that?
Yeah, so to be honest, this is not anything that I know anything about. That's a totally different area of astronomy. But definitely the type of star is going to change the Goldilocks zone quite a bit. I mean, part of the reason that things work so well for us is that the sun is kind of a medium temperature star, medium size. It's not very active. And so as soon as you start getting bigger to stars like what I study, they are very massive stars. They're much hotter in many cases. And so that's going to change things a lot. And then also a lot of stars have really strong magnetic fields that very much hinder any sort of life forming on any planet. And so definitely the type of star very strongly dictates kind of if planets can even form and then survive around them.
Awesome. Last question. What would happen to our Earth if the sun was replaced by a red supergiant?
Well, we wouldn't exist. So if you just kind of like suddenly replaced it, in most cases, our Earth would be consumed by it. The red supergiant would extend out past the Earth. And so certainly in the case of a red supergiant, the Earth would go away very quickly. And eventually the sun will expand not into a red supergiant, but into a red giant. So it'll also cool down and expand. And humans probably won't be alive at that point, but something similar. I mean, not a good outlook for the Earth in that case.
Okay, sounds good. So let's just hope that doesn't happen. Thank you so much for sharing that with us. That was super cool. Yep. And you already shared where we can find you online, so we are all good. Thank you so much.
Yeah.
At this point, Karen, you're up.
Hello.
Hello.
Let me just get my screen up really quick. share my screen. Okay, you can all see that?
Yes, yes.
Great. Okay, so hi, my name is Taryn Black. I'm and I study glaciers in Greenland and how they are responding to climate change. So while we're all sitting at home, I'd like to invite you all to think about where, if you weren't home right now, if you could actually travel somewhere, where would you like to go? And I imagine a lot of people are probably thinking tropical beaches or a bustling city or more tropical beaches. But I personally really kind of prefer colder climates and more rugged terrain. And I would love to go to Greenland, specifically to this town that's called Ilulissat in Greenland. It is on the west coast of Greenland. So you can see this map of Greenland up in the upper right corner here. And what you might notice is in the background, there's a lot of what appears to be ice in this picture. And it is in fact ice. Those are probably big icebergs that are sitting in the water just off the coast of this town. And they're coming from a glacier that's called Jakobshavn. So Jakobshavn Isbrae, Isbrae just means glacier or ice stream in Danish, is a very large glacier on the west coast of Greenland, really close to this town of Ilulissat. The Greenlandic name is Sirmak Kujalek, which simply means southern glacier because it's in the south. And this glacier is one of the largest glaciers in Greenland. It's one of the fastest flowing glaciers on Earth. It contains enough ice that if it were to all melt, it could raise sea level rise rather substantially. And it creates a lot of very large icebergs. It's even thought that this is the glacier that created the iceberg that sunk the Titanic. So this glacier is really big. You might not be able to get a sense of scale from this photo, but I've put this Statue of Liberty here for scale. And the Statue of Liberty is about 300 feet tall from the base of the pedestal to the top of the torch. And that's about how tall this glacier is just from the water level up to the top. There's even more ice beneath the water level. So these are massive, massive, massive glaciers that we're looking at here. One more thing I'll point out is that this photo is taken from an airplane, which is a pretty cool close-up view, but I am more used to looking at glaciers from this kind of view. This is a satellite view of the same glacier, Jakobshavn. This image was actually just taken yesterday by a satellite called Sentinel-2 that's from the European Space Agency. I'll just give a plug for free public satellite images. There are a lot of different places you can find them, but I usually use this site called Glovis from the United States Geological Survey. I find it really easy to find images there. So this is the typical view that I have of glaciers because I study them using satellite images. So what we have, and the town of Ilulissat is just on the left here to get you oriented. So what we have here, I'll try and turn on my laser. There's a lot of rock with a dusting of snow on it, kind of on the left and in the center of this image. The ocean is on the very far left. You can see that very dark color is actually open ocean, and the little white spots on it are little icebergs. As we come into this big fjord here, this big long channel where there's water, there's a lot more ice floating on top of the water. And then on the far right of the image is the actual ice, the glacier and the ice sheet itself. So what I'm typically looking at in this kind of satellite imagery is I want to know where the front of the glacier is. We call that the terminus, and I would trace it in this picture, it would be in this position. So I use satellite images to study how glaciers in Greenland are changing. I want to understand how much they're changing and how fast they're changing and what factors are causing these changes. So in order to understand that, we need to first quickly think about what makes a glacier in the first place. So a glacier is a place where snow turns into ice. That ice sticks around year to year, so it doesn't completely disappear every year. It's actually there for the entire year round. And there's enough ice that it's actually heavy enough that it flows under its own weight. So every single snowflake that falls on a glacier, unless it perhaps melts, every snowflake that falls is going to be compacted. Like if you took a handful of snow and made a snowball, you squish it down and it almost turns into an ice ball. It's really hard. That's what's happening on a glacier. All of these snowflakes that fall get compacted into ice, and very slowly over time, they will move down the glacier, down and down and down, until they get somewhere where they might eventually disappear. So the ways that we lose ice on glaciers are through melting, for example, on the surface. So this is a picture of a melt pond on the surface of the Greenland ice sheet. And we can also lose ice on these glaciers in Greenland and some glaciers in Antarctica. We can lose ice through what's called iceberg discharge. And this is the process by which ice breaks off the end of a glacier and forms an iceberg and floats away in the ocean. So this is a really cool video that you can find on YouTube of a glacier called Helheim in Greenland. It's on the east coast of Greenland. And that was a massive, massive iceberg that just came off of this glacier. It's so impressive how big these things are. So these processes determine what we call the mass balance of a glacier. On one side, you have addition of ice to the glacier through processes like snowfall. And then on the other side, you lose ice through melting and icebergs. So if these two processes are in balance, then your glacier will more or less stay the same size. But you can also have what we call a positive mass balance, which means that you get more ice gained than you've lost. If your mass balance is positive, then your glacier is going to grow because it's getting more ice faster than it's losing it. So the front of the glacier will advance. If you have a negative mass balance, that means that you're losing more ice than you're gaining. And so the glacier is going to shrink and the front of the glacier will retreat. So we can observe these processes seasonally. What I'm showing on the left here is a GIF, a series of satellite images of, again, Jakobshavn Glacier on the west coast of Greenland. And we're just looking at it every week or so for a couple years, and you can watch how the front of the glacier that was circled there for a minute kind of moves forward and moves back a little bit throughout the year. On the right side, what we're plotting is how the length of the glacier is changing in each of these images. And it'll be done in a second here, but what you'll generally notice with this trend is that the glacier retreats more in the summer when it's warmer, and so it's not snowing very much, but it's losing a lot more ice. And then it tends to advance more in the winter. When it's colder, it's snowing a lot, and it's probably not melting and making as many icebergs. So we can observe these processes seasonally and see these kind of small ups and downs in the mass balance throughout the year. But we also can watch how mass balance plays out over many years. So here is a picture, a satellite image of Jakobshavn from 1972. This is one of the earliest satellite images that I actually have of this glacier because we didn't have very many satellites up much longer before that. And this is an image from 2018. It's from a slightly different type of satellite, which is why it looks maybe a little bit funny compared to the other image. And you can see that the front, which I've traced in red, is in two different locations here. And we can overlay them on top of each other to look at how much ice Jakobshavn has lost in the last 45 years. So again, we have the 1972 position and then the 2017 position. You can see they're a bit far apart from each other, but how far is that? That's actually 8 miles. That's a pretty substantial retreat for this glacier. And again, my job is looking to see how much these glaciers have changed. So maybe I can look at this and say, all right, wipe my hands, I'm done because I figured out how much this glacier changed. But is that sufficient? Is that enough data? We know where it was a long time ago, and we know where it is now, or a couple of years ago, and we can measure how much it changed. But that's kind of like saying that the Seattle music scene, I'm from Seattle, by the way, so this is like saying that the Seattle music scene had heart, and then it had Brandi Carlile, and that's it. There's no other music from Seattle. Seattle is not known for any other kind of music whatsoever. You might see where I'm going with this. This is not sufficient. We want to look at 45 years of change at Jakobshavn, not how much has changed just a first and last measurement, but how it's changed throughout those years. So here's our 1972 and our 2017 measurements again. And I'm gonna add all of the other fronts for every year between 1972 and 2017. So now we can look at this glacier and say, okay, yes, it did retreat, it got smaller, but, there we go. We can see when we're looking in more detail, we can see that in the first couple decades, it wiggled around a bit, but it didn't retreat very far. In the late 1990s and early 2000s, it retreated very fast and very far. And then in the 2010s, it slowed down and it started kind of just wiggling around in the same location again. So we can again ask, is this sufficient? We've gotten a more detailed record of how the size of this glacier has been changing. We've got our Hart and our Brandy Carlisle, and we've also found Nirvana. But this is just one glacier, and this is really helpful for trying to understand specifically how Jakobshavn is behaving, but I want to understand how all of the glaciers in Greenland are changing and what's causing all of them to change. So Seattle is probably not the only music scene that's out there, right? There are other bands in other countries, other states, other continents that have created a lot of music. So we need to look at those as well to get a better picture of what's going on. So to that end, I've built a large data set of glacier front positions in northwestern Greenland. I've looked at 87 different glaciers over more than 45 years, and I've individually traced by hand over 3,600 individual glacier fronts. I'll also make a note really quick. The data that I've been showing goes to 2017. I do have more current data, but thanks to COVID, I've been having some serious internet issues recently, and I actually could not download the data to update my plots for this talk. So rest assured, there are still satellites up there. I am still watching what's going on with these glaciers, but I couldn't update the figures for this talk, unfortunately. So from all that data that I've put together, I've found that Northwest Greenland's glaciers are retreating. All of them are retreating. This is the total area of all of these glaciers from 1972 to the present. And you can see that the combined area of all of these glaciers is getting smaller. In the first few decades, the ice loss was a little bit slower, and in the late 1990s, it got faster. This shows us that the mass balance of all of these glaciers is out of whack. They all have negative mass balance. So what's going on? Why do we see this widespread retreat of all of the glaciers in northwestern Greenland? They're all retreating, and they're all retreating faster around the same time. I want to understand what is causing this, so I looked into several different factors. And if we think about our balance again, it might be that there's less ice available, so that as these glaciers are losing ice, they're not getting replenished by more snow. Or it could be that they're losing ice faster, they're losing more ice. So there's many different factors that could be causing that, such as snowfall or air temperature, ocean temperature, the rate of iceberg production, all kinds of things. So I looked into a lot of these and compared them to my record of glacier change. We actually found that sea ice and melange seem to be more important than a lot of those other factors that I listed. Sea ice is frozen ice. Sea ice is what happens when you freeze ocean water. So it's ice that forms just on the surface of the ocean. It's kind of salty. And then melange is pieces of fresh ice that have broken off of a glacier, kind of like little icebergs, and they're just floating on the surface of the water, and they can freeze up with that sea ice during part of the year. So if sea ice forms on the surface of the water, it'll freeze all of those melange bits, icy bits in place. So how could this be important for why our glaciers are changing? How would this matter? There are several ideas out there about how this could work, but one idea that my advisor and I have been working on is this idea that sea ice and melange can provide some resistance to iceberg production. So, for example, if you have rigid sea ice in front of a glacier, that can slow the creation of icebergs. It's kind of like putting a cork in front of the glacier, in the fjord in front of the glacier. There's something pushing back it's just applying just enough pressure to keep more icebergs from falling off of that glacier and floating away. But over the last couple decades, we found that there's a lot less sea ice and melange than there used to be, and it's not as strong. There's been less forming each year, and it doesn't last for as much of the year. So there's still a little bit of pressure pushing back on that glacier, but it's not as much as it used to be. And we think that that could be allowing more icebergs to form for more of the year, which is causing these glaciers to lose ice faster. So just in summary, glaciers in Northwest Greenland are shrinking, and that ice loss sped up in the late 1990s. Because all of these glaciers are behaving the same way, we think that there's a regional cause for that, and our most recent research suggests that that might be due to seasonal changes in sea ice and melange. So this will help us better understand how these glaciers are behaving now and how they might behave in the future as they continue to respond to climate change. Thank you. I'm happy to take any questions.
Thank you so much. That was great. Okay, that was so interesting. We've got a couple questions for you here. Okay, so how large does a sheet of ice need to be before it's officially considered a glacier?
Oh, that's a good question. I know a lot of people who think about this in the reverse right now, that how small does a glacier have to get before it's not considered a glacier anymore. There is actually some recent media about a glacier in Iceland that got demoted to just a chunk of ice. They had a funeral for it and everything. I don't think there's actually a consensus on that question. Even among glaciologists, we kind of loosely define them as ice that persists throughout the year and can flow under its own weight. But that brings up questions of what kind of time span you're looking at, and does it have to flow over the entire year or just part of the year, and so on.
Cool, thanks. And so have there been times in Earth's history where there have been absolutely no glaciers at all? And are we on a path to going back to that if that ever happened?
Yeah, there absolutely have been times in Earth's history when there hasn't been any ice on the planet. That's actually, I guess I couldn't say what percentage of Earth's history that is, but it's decently common. The term ice age actually just means that there is ice at the poles. So in the times of the dinosaurs, and even after the dinosaurs for a while, there wasn't any ice on the planet, and it's now gotten cold enough that we do have not only ice at the poles, but these very large ice sheets that grow and shrink as we have these glaciations that send ice down over Seattle and so on. And I'm sorry, remind me the second part of your question.
Well, I'll pivot to this question.
Okay.
Typically in Earth's history, things move kind of slow. Like things change slower than they're changing now. So what is the difference in like the results basically that we see globally in a slowly dripping, like dissolving glacier versus really quickly everything melting?
Oh yeah, I gotcha. Oh, right. And your other question was, are we on that path now?
Yeah.
Yeah. So for both of those, we're not going to see no ice on the planet in our lifetimes or our grandchildren's lifetimes. I think it would take quite a bit to do that, but we definitely are on a track for substantially reducing the amount of ice that we have on Earth. I think it is not out of the question that we could lose a lot of the ice in Antarctica because it's kind of near a tipping point. And as to your other question about timescales, that is a good question. Ice definitely changes a lot faster than a lot of other geological processes, and I'm kind of in two worlds of having a background in geology and thinking about those very slow processes, but the thing that I study is much faster than that. Yeah, I don't think I really have a great answer to that, actually. I'm sorry.
That's okay. That's okay. Saying you don't know is very important. And last question, who or what is your inspiration and when did you get into glaciers?
Oh, wow. Okay. Trying to get the short version of this answer. So I had a fantastic 8th grade science teacher who really got me into physical science. Followed by another fantastic chemistry teacher in high school who got me interested specifically in geology. He was a geology enthusiast. I also joke that I worked at an ice cream store in high school, and that was actually my introduction to working in polar sciences, because after that, I found a research job on campus when I was an undergrad. I saw an ad that someone needed someone to work in a freezer handling ice cores. And I was like, oh, I've been working in a freezer for 5 years. I'm so qualified for this. So that was how I started doing polar science. And I've had a bit of a meandering route after that, but I got really interested in studying climate and glaciers and climate change. So that's kind of how I ended up here.
Awesome. Thank you so much. Okay, is there anything else you'd like to plug before we go on to our last talk?
So I had my Twitter handle on my slides, which I've now closed, but you're welcome to follow me on Twitter. It's Taryn E. Black. And I do also want to plug, I'm part of a science communication program at UW that is also doing a bunch of live streamed talks right now. So if anyone's interested in that, you can look up Engage Science on Facebook or Twitter, and we'll be posting links for our upcoming events.
Awesome. Thank you so much. Okay,
thanks.
You're welcome. Last up, we've got Vitika.
Let's see. I'm getting set up. All right. Let me get this set up real quick here. All right. So can you see my first slide here with
Looks
beautiful
good.
albatross? All right, awesome. All right. So aloha, my name is Vitica Holtheisen, and I'm a master's student at Northern Illinois University, and I study poop. So when I start talking about poop, people sometimes get a little bit confused or uncomfortable. And as much as I love SpongeBob, I'm actually not talking about people ordering our patties. I'm actually talking about real poop. And to be honest, I never really had thought much about poop before in my life. For years, I studied birds, actually, all across the United States, from backcountry of Arizona to the Missouri Ozarks. And then I went to Midway Atoll National Wildlife Refuge to help study seabirds and their population trends. And I had something happen to me. This tiny Laysan albatross chick in the photo did a massive poop on me, and that changed the course of my life. So I came to learn actually that seabirds poop, and they poop a lot. And if you've ever had the privilege of going to a seabird colony, you'll notice two things. So one, you'll see that wherever there's birds, there's lots of bright white streaks on the ground, and that's poop. That's their poop. And you'll probably also notice a very pungent smell. That's also the poop, or rather what's called guano, which is actually accumulated poop along with even eggshells and carcasses that over time forms a natural mineral deposit. And this guano is just chock full of nitrogen, phosphate, and potassium, all of which are really critical nutrients for plant growth. So why should we care about seabirds and their poop? Well, because seabirds tend to nest on small, remote, and isolated islands, they're actually really pivotal to supporting entire island ecosystems by bringing in nutrients that support not just plants, but insects, amphibians, and lots of other wildlife. So to give you a better idea of this, here's a little diagram. So here we have our seabirds, which poop on the island. This poop fertilizes plants, which provide important breeding or nesting sites, but also provide food and cover for other island wildlife. Some of these guano nutrients also move through the soil and actually go into the waters surrounding islands where these nutrients support healthy algae, corals, plankton, and fish. And then seabirds eat these fish and the cycle continues. So essentially on these seabird islands, guano acts like a fertilizer, and that attracted people. Many believed it would revolutionize farming, which traditionally involved cycling crops or simply depleting soil nutrients and then moving on to new land. Guano changed the farming game. Seabird islands all over the world were claimed and heavily mined for guano. And unfortunately, in the process, many of the seabird populations on these islands declined dramatically because of human disturbance or just went extinct altogether. Then throughout the late 19th and 20th centuries, people continued to explore and exploit these seabird islands for lots of other purposes, such as collecting seabird eggs for albumin, which is the egg white, which was used for photo development, seabird feathers for decorating clothing and for the feather trade, and even wartime installations. So one such seabird island is that of Midway Atoll, which is here, and it's very aptly named. It's a tiny set of islands smack dab in the middle of the North Pacific Ocean, equidistant from Japan and the west coast of the United States. And I live here in DeKalb, Illinois, which is almost 5,000 miles away. Midway, which is circled here in orange, is also known in Hawaiian as Piheimanu, which is the loud din of birds, or Kauai Helani, which is backbone of the heavens. And it's located nearly at the end of the northwestern Hawaiian Islands, which are part of a much larger Papahanaumokuakea Marine National Monument. These islands are part of a really long chain that extends over 1,200 miles northwest of the main Hawaiian Islands, right here. And although Midway is small with its tiny islands covering only 2.4 square miles, there's a lot of life out there. In fact, Midway is home to the largest colony of albatross in the world. Over 1 million Laysan albatross, which are pictured here, breed on Midway each year, and it's amazing. This photo captures just how dense life is on these kind of seabird islands. In total, more than 3 million birds from over 20 different species breed on Midway or use it as an important stopover point, like a resting or refueling station, during migration. So even as a speck in the ocean, it is teeming with life on Midway. But Midway didn't always look like this. Because of its location, Midway was a prime spot for wartime efforts, and as a result, during both World War II and the Cold War, much of the island was paved over and altered. Nests and even the birds sitting on them were bulldozed over during construction, and valuable habitat was destroyed. Birds collided with planes, overhead wires, and other hazards. And in addition to the planes and bombs and people, lots of new things came to Midway that weren't there before. To support the growing human population on Midway, lots of agricultural crops were brought in. People also brought along reminders of home, like their favorite flowers. And in the process of bringing in all these materials and supplies, a couple of hitchhikers also tagged along. One of them being house mice. Yep, the same kind of mice that you actually might have in your own home. But these house mice are killer mice. And you're probably thinking, yeah, whatever. No, but for real. Starting in 2015, house mice on Midway Atoll began to attack and even kill adult Laysan albatross. Well, how does a tiny mouse take down a huge albatross? That's crazy. Well, think about this. Albatross, like many seabirds, evolved in isolated environments absent of mammalian predators. So consequently, albatross don't have a very good sense of danger, so to say, nor do they really have strong anti-predator behaviors. So in order to protect Midway as a globally significant breeding area for seabirds, mice will be eradicated next year in 2021. To eradicate mice, the Fish and Wildlife Service and the nonprofit group Island Conservation will use a rodent-specific poison. Oh, my bad, wrong poison. Great song by Alice Cooper, though. They're actually going to use a special rodenticide called Verdificum for the mouse eradication. So this seems like an easy fix. Like, mice are introduced invasive predators. They're having negative impacts on Midway seabirds. So get rid of them, right? Well, things are never as simple as they seem, even on islands, which are pretty small and seemingly simple ecosystems. Introducing exotic wildlife or removing this wildlife from islands can have unintended consequences. What do we mean by that? Well, we learned that mice on Midway only attack albatross during the winter months. So they're eating something else throughout the year. What that is, we don't know. Mice are omnivorous, so they eat a wide array of things like plants, seeds, and insects. And even with an eradication, we're unsure of what the consequences will be. Do mice eat invasive plants? Do mice eat pest insects? How will these organisms respond once mice are gone? So we know that mice affect their environment through what they eat. So to figure out how Midway might change after the eradication, we need to figure out the diet of Midway's mice now. And this is where poop comes in, and hair. By using next generation sequencing and advanced DNA analysis technique, we can uncover the DNA of diet items in mouse fecal samples. Basically, this gives us a list of what mice are eating. In other words, it's a menu. But if we want to understand how much mice are eating in terms of different diet items, we use something called stable isotope analysis. The idea here is that you are what you eat. The nitrogen and carbon of what you eat is absorbed into your body and assimilated into tissues like blood, bone, and hair. By examining the stable isotopes of both mouse hair and common mouse diet items, we can figure out how much these diet items contribute to diet as a whole. All right, so big moment. What are mice on Midway eating? We're still learning, but here are some of our preliminary findings. So first of all, plants are virtually absent from mouse diet. That's a surprise. Insects do contribute to mouse diet, but much less than we'd expect. And most surprisingly, there's a lot of seabird tissue in mouse diet, much more than expected. And it also looks like seabird tissue is present in mouse diet year-round. We know that mice on Midway have attacked albatross in the winter, but we haven't seen attacks during any other season. So maybe mice are scavenging seabird carcasses. With this information, land managers can prepare for ecological changes post-mouse eradication. Plus, we can track how affected plants and wildlife will respond to mouse eradication. In turn, our research can help to guide the ongoing recovery of Midway's ecosystem. And ultimately, our research can help to guide future mass eradications and ensure conservation success on other islands. So with that, I'll end my presentation. Thank you so much for tuning in, and be sure to follow me on Twitter to learn more about Midway. Mahalo. All right.
Okay. Thank you so much. This was great. So here's a question. How long did it take for birds to rebound and re-enter that area after the war?
So, I mean, there were still many albatross that were out on Midway after the war. Unfortunately, we don't have like fantastic data on how many albatross there were beforehand, but we know that their population has definitely rebounded quite substantially. We've seen some record counts in the last several years, so we know that for albatross, at least, they've recovered very well. There are also several other bird species like red-footed, brown, and masked boobies that have been a little bit slower to recover because they also suffered from impacts from other invasive mammals on Midway, like rats, but they were thankfully eradicated in the mid-1990s. So on the whole, Midway's avifauna, or all the birds that are there, have responded pretty well and relatively rapidly.
Awesome. So how many birds species live there, and how many of those bird species are endangered?
Great question. So there's over 20 different species that breed on Midway or use it as a migration stopover point. There's a couple of different endangered and threatened species. One of the endangered species, actually two endangered species that are out there, include the short-tailed albatross. There's one pair that just had a chick this year. There's also lace-end ducks, which are arguably the most endangered duck species in the world, that are on Midway. And then we have two threatened species, the black-footed albatross, and I believe the bristle-thighed curlew.
Very cool. So what would a healthy or totally recovered post-mouse ecosystem look like?
It's a fantastic question. Well, I think the nearest example that we could think of are other islands in the northwestern Hawaiian Islands that are also really large seabird colonies where you don't have invasive mammals. So good examples are Kure Atoll and Laysan Island. These are really enormous, robust, and bustling seabird colonies, and we would hope the same for Midway as well.
Awesome. Thank you so much. Okay,
Thank
so
you.
let's bring everybody back together for wrapping this up. This was so cool. I learned so much tonight. This was awesome. You all gave such wonderful presentations. And I loved hearing about all your research. Is there anything else that you'd like to share with everybody before we wrap up? No? Okay, awesome. So thank you everybody for joining us today. This was really, really fun and awesome. Scientists, I'm going to be sending an email to you soon to think about how we could do this again and be better next time. But everybody else, thank you for being here. We are Skype of Scientists running this program. And if you're interested in stuff like this, what you can do is check out our website, skypeofscientist.com. Click on the live streams tab, and you'll see all the stuff that we have going on in the coming months. We do about 3 live streams a week with one scientist for 45 minutes, and there's a lot more time there for specific questions. But this was just so fun, and thank you all for coming. And that's it. I'll see you guys later. Bye.